everyone, and welcome to the National Research Collaborative Meeting 2020. Thank you for joining us. We're very excited to present our webinar, Research Red Tape: What can we learn from undertaking research in a pandemic? We have the amazing opportunity to hear from some of the orchestrators of the recovery study and the COVID Surge Collaborative. Before we start, I'd just like to go through some simple housekeeping. On the left-hand side of the screen, you'll see a reception tab, a stage tab, and a sessions tab. Our schedule and poster and video gallery is available in the reception area. Please ensure you have a look at the high-quality research being conducted around the country. Please also fill in our poll, which can be found in the polls tab, to indicate which field you work in. Our speakers are hosted in the stage tab. Please use the stage chat for questions only, and any other questions or conversation should be held in the event-wide chat. After the event, we'll have coffee shops, which will be hosted in the sessions tab. Please join us there for conversations about tonight's content. And after the event, we'll be sending feedback links. After completing it, you'll be offered a certificate of attendance. Just make sure Generate an Attendance Certificate is toggled at the bottom of the form. Now, I'm pleased to introduce our chair for the evening. Jane Blaisby is a Professor of Surgery at the University of Bristol. She's the Director of the Bristol Centre for Surgical Research, a multidisciplinary centre whose overall aim is to design, develop and deliver studies that evaluate surgical interventions to inform evidence-based practice. Over to you.
Thank you very much, Emily, and welcome. I'll add my welcome to everybody to this NRCM meeting, which we had hoped to have you all coming here for a drink in Bristol, but we very much hope that we'll be able to do that face-to-face next year. It's a real privilege to be able to chair these two projects for you all and talk to you all about them this evening and hear more about them, because the Birmingham team who've led the COVID Surge project have worked so brilliantly over the years, and I'm so delighted that Martin Landry can join us from the recovery trial, which clearly we are all individually very grateful to have run from the UK and with such fantastic results for the whole world. So it's very exciting. So I'm going to hand over, first of all, to James Blaisby and to Elizabeth and Joanna, James is going to present just the highlights of the study, and then they're going to show a video about how on earth they managed to so quickly and so efficiently and so well run the important COVID Surge study. Thank you, James.
Thank you very much, Jane. It's a pleasure for the three of us to be here this evening. So we've been told strictly not to focus too much on the COVID Surge sort of nitty-gritty results today, and we're going to try and give you an overview of the process and how things got set up quite swiftly at the start of the pandemic, and give you a little bit of a insight into how these big international studies can run at scale. So we've got a little elevator pitch for you first, just to run over just maybe the top 5 lessons we've learned from COVID Surge. I think the first thing to say is that this is not our work, but work that's owned by thousands of clinicians, surgeons, trainees, anaesthetists, all around the world. And the reach of this network has now expanded to including over 150,000 patients across the study portfolio, with over 15,000 surgeons. So that's about 3 times larger than the large Hadron Collider collaborating group. So this is the largest collaborating group we think ever put together for a scientific study, and without the work of every single individual within this, it really wouldn't have been possible. The other sort of very important group of people to thank for the work that COVID Surge has done really is the funders, and it's a really tricky year for charities and funding organisations. Everyone's focusing on COVID, but these organisations put themselves out there and supported us at a really crucial time at the beginning of the pandemic. So a big thank you to all of these groups. So quickly, elevator pitch number 1, there was a huge amount of elective cancellation of surgery around the world in the first phase of the pandemic. And we estimated in a modelling study that about 30 million operations were cancelled in the first 3 months of the pandemic. And this will be expanding further and further still. And the reason was, of course, a worry about the risk of SARS-CoV-2, the coronavirus infection, in the perioperative setting. So over to Lizzie to give us the elevator pitch about the COVID Surge Lancet paper.
So, hello. So after that, there was a real need to understand how this novel coronavirus affected patients, and a need to gather patient-level, high-quality prospective data. So we started this cohort study, and firstly looking at patients with perioperative SARS-CoV-2 diagnosis, so 7 days before or 30 days after. And what we found in the first 1,000 patients, which was published in The Lancet, demonstrated that of these patients, there was a very high mortality rate of 24% compared to previously cited surgical mortality rates. And of these patients, 51% developed a post-operative pulmonary complication, which in itself had a 38% chance of a 30-day mortality. The patients most at risk were male, they were elderly over 70, they were sicker of ASA grades 3 to 5, cancer diagnosis and surgery was more risky, and emergency and major surgery. And our conclusion after the first analysis was that post-operative complications and mortality is higher in these specific groups, and that these patients should be protected.
Thanks, Lizzie. So then we know that SARS-CoV-2 in the perioperative setting was really severe. So our second study, which was called COVID Surge Cancer, well, this included patients undergoing elective cancer surgery, that protected cancer, surgery type during the early phases of the pandemic. And we looked first at the operated patients and ways that surgeons around the world were trying to protect patients from SARS-CoV-2 infection. And the first thing we examined was the role of these COVID-free or COVID-light hospitals that we've seen being set up. And we asked, did these kind of COVID-free surgical pathways actually protect patients? Because they're a huge headache, huge administrative effort, and take a large amount of funding. And what we showed in this paper that was published in the JCO a couple of months ago now, we demonstrated that there was this consistent evidence of COVID-19 free surgical pathways reducing the risk of infection and subsequent pulmonary complications and death. And this worked whether or not the community rate was high as it is now or low. There was still this benefit that we saw. So that was the sort of next phase of the project, saying, you should set up COVID-19 free pathways to encourage surgical safety. And the next point in the story, I'll pass you on to Jo for our third elevator pitch.
Thank you, James. My name is Joanna. I'm also one of the research fellows working in the collaborative. So from this study about cancer patients being operated during the pandemic, one of the research questions that the surgical community had during the first wave, and now as well, is which testing should we be doing to kind of mitigate against asymptomatic carriers to carry the virus and have pulmonary complications after surgery, which we have seen that carry a high risk of mortality after surgery. So we've compared different testing strategies. We've compared patients undergoing swab testing, CT, both or no testing. And we've come to the conclusion, both in the adjusted and non-adjusted analysis, that actually swab testing is the one that is better correlated with lower pulmonary complications after surgery. So the recommendation is actually that swab testing is the best way to prevent asymptomatic carriers to carry the virus and have complications after surgery. And we've done some sensitivity analysis just for the patients being swabbed, and we've calculated the number needed to treat, comparing patients undergoing minor and major surgery and in high and low incidence of COVID areas. And we've came to the conclusion that swab testing is relevant overall, but more important when patients are undergoing major surgery, especially when COVID, the community incidence of COVID is high, and the cutoff for this was more than 25 cases per 100,000 population. So this is one of the research questions that we also took from the cancer study.
Thanks, Jo. So we've heard the story. Lots of operations cancelled because of this fear of perioperative infection, but we think that this data gives the evidence needed to upscale surgery safely through COVID-19 free surgical pathways and routine preoperative testing. And I'd encourage you all to download our post-pandemic recovery report from this link, which details in Technicolor glory, curated by Dawood, and he's a Birmingham medical student, and Connor Jones, he's a trainee in Bristol. It's a very nice report showing sort of an overview of all of the data. So without further ado, we've done a little fireside chat with the three of us and Anil Bangi, who's the study chief investigator. And we'll, if it's okay, Phil, we'll go over to the videotape to watch that back.
Do put questions in the stage chat, anything as we go along.
Hello, and thanks for joining us today. It's a pleasure to be here for this year's National Research Collaborative meeting. And we're going PowerPoint free, so we're going to do a discussion amongst the central COVID surge management team who are here about what went well and how we got it going so quickly, and where we can improve and what the future vision is. So my name is Anil Bangi, and I'm a consultant colorectal surgeon at University Hospital Birmingham. And I'm a senior lecturer and clinician scientist. And I sort of grew up through the National Collaborative, which has really got me to where I am. So I'll let the rest of the team introduce themselves. So let's start with Elizabeth.
Hi, I'm Elizabeth Lizzie. I'm a surgical registrar and NIHR doctoral research fellow in surgical innovation, and again also involved in the collaborative research from a SHO level, and really excited to be here.
And over to Joanna.
Hello, my name is Joanna. I'm a general surgery trainee from Portugal, part of the PT Surge Collaborative as well, and now doing research in the Global Surgery Unit in Birmingham. Glad to be here today.
And last but not least is James.
Hi, everybody. I'm James. I'm, again, a doctoral research fellow in global surgery, working in Birmingham. I've been involved in Star Surge for a while to cut my teeth, and now doing some global surgery work with Anil and the team internationally.
So it's been a tricky year for all of us, and I think we're all aware that trainees have been on a particular roller coaster with COVID rotas, changes in training, and we feel that, and we feel for what's happened to the patients as well. But you know, from my point of view, the trainees have shone in the research and the COVID surge side, and they've produced this data which has been pretty amazing, and has gone around the world. So let's just explore that. Let's start with Joanna. Joanna, if you think back to March, what's your take on how this started and how you got it all up and running so quickly?
It was actually quite an amazing setup in terms of how could we get a lot of efforts together to set up a project so quickly. And I think one of the main things there is that because everyone was very focused on this single project, and for a while there were no side projects, and all our attention and efforts were towards this particular project. And obviously you can't simulate and replicate those conditions for all of your projects, but you might learn from that, and you might want to in future projects for a particular time window, focus your team on something and kind of put all the projects in standby so you can get networks and projects set up really quickly.
To flip it just to the other angle, James, just look at this from the other side. So you and I went onto these COVID rotas, didn't we, in March, April, May, and a bit of June. And I think most of the people listening today probably ended up on some form of COVID rota, and they were emotionally tough. How did you balance doing all of this COVID surge work with being on a COVID rota?
Thanks, Anil. Yeah, as you say, it's tough for everyone. I think that has been working in the COVID environment. It was quite a stressful time and exhausting for people. But actually, these collaborative data projects were something really positive, which came out of all of the chaos around COVID. I think it really built a community, and people felt part of something. And it was something which they could, take home and say that this was their part in a broader COVID effort. So I think actually spending some time outside of work on this, I think was really positive. And certainly that's the sentiment I've heard shared by others too.
And Lizzie, do you think, taking those two things into account, that people were busy and tired, but people wanted to take part in something, do you think that contributed to the range of specialties who took part in COVID surge? Because you might want to summarise, but I think we had representation from every single specialty.
Yeah, we definitely did. There's probably was across all specialities a degree of almost nervous energy as this unforeseen wave started like sweeping the entire world, and all of our lives and working lives were changed, and people were looking for something productive and positive to channel their energy into, and also to get some answers as well. And it certainly did unite the entire surgical community because we were all afflicted by this, and I think that very much helped us answer these critical questions and get COVID surge off the ground.
And so on that point, we'll flip out. We'll go backwards. So James, I'd like to hear from all of you on this. This is about the design phase. So it was designed very quickly. The first thing I'd say is we went cross-specialty. Would you recommend people to do cross-specialty projects, or do you think that is too challenging? And bearing in mind that you might be talking to people who are working in smaller teams.
No, it's a really good point. So I think in general, what we've learned from this and previous projects is actually the kind of bigger ticket topics which cross over multiple facets of surgical specialities, and perhaps lean into medical specialities, critical care, anesthetics. Actually finding those borders between overlapping Venn diagrams is a real strength, and means that the research we create is meaningful to more patients. So certainly cross-speciality working, I think, is a real strength. In COVID surge cancer, which was sort of a parallel project, we worked with leaders in lots of different specialty areas to design kind of bespoke additional parts of CRFs that could give specialty specific insight. And that did have its challenges. That's a lot of data to handle and curate and to get right. But I do think that kind of cross speciality method of working is something we could definitely learn from for the future.
Maybe on that, Anil, we can also say that in the design phase, it was quite productive and quite meaningful for the design phase to have feedback from collaborators, not only leads, but actually collaborators from different countries, different settings, different specialties. And when you go back with your ideas, your rough ideas to your collaborators, you do get a lot of information back and feedback. There is always a bit of noise when you do that, but you do get an idea of what is relevant to a broad group of people. And actually you design the research questions for your outputs later on to be more generalizable in terms of what they can influence in clinical practice.
And so Joe, to come back to you on that, you're talking about the design phase, and you've had all this information back to people. I'm also thinking of our Falcon trial, which is closed and has closed in record time with really high data completeness. Harnessing that all together, I think we had one of the most efficient design processes for our protocol and our CRF, which are the report forms people fit in. From your point of view, how were we so efficient in a protocol and a CRF over such a complex project?
So I think it probably takes a lot of responsiveness between the team designing the CRF, being able to absorb the feedback, get back to the collaborators again. So this kind of short-term, go back and forth with your project kind of speeds you up and allows you to have a completed or full CRF quite quickly. And also, I would say that one of the main things is that I'll highlight it to people, because this was always going to be a large scale, global scale study, was please do make sure that we are only including data points that we are actually going to use. So trying to make our CRFs as short as possible and as directed to the research questions as possible was also very important to kind of move forwards quickly.
So Lizzie, we sort of poured over, if we look at the COVID surge project, the first one, we spent a lot of time producing a very short protocol, and every data point that went onto the CRF, we sort of focused it down and figured out if we needed it or wanted it. Do you think, would you recommend other people to do that, or is that specific to this project?
It does depend on what you're aiming to do, but in general, with our experience, erring on the side of simplicity, designing it to answer the question you set out, not to broadly try and gather as much information as possible, and also ease of use for your collaborators. And also, as Joe and James have alluded to, listening to the feedback from our collaborators as well, those on the front line, what are the questions they need asking? So amalgamating all that together to answer just the critical questions was what we found the most successful model.
On Lizzie's point, I guess one of the other things we did for the first time here was to build some like flexibility into the protocol. So we were able to kind of change the protocol, change the CRFs, and we built that, you know, we wrote that into the protocol that this would be responsive to new changes and adaptation. And that meant that we had like a formal second phase of the project where we introduced some new data points and we took some out that weren't working. And it's the whole kind of fail fast, fail early. Once you've got started, it's much more easy to learn from experience. So perhaps that's another useful lesson for the future.
I'm going to ask you each a specific question about delivery. So Joanna, Dissemination Committee, what was it? What is it? And is it useful?
So I think it was the first time we've probably gave it that name, Dissemination Committee, but you always have this group of people that kind of buy into your project and kind of disseminate, you know, spread the word about it and everything. In this project, because we wanted to go big scale and global scale, we thought about this Dissemination Committee by being a group of people who would be leading their own countries, setting up for the study, but we made it very flexible and inclusive. So for instance, we didn't restrict it for one single lead per country. If people could get help from other parts of the country to get the whole country involved, for instance, we would allow more people to join within, let's say, Germany or South Africa or any country. And this Dissemination Committee also included people who would be representatives from international societies who could work across different countries and disseminate the studies and the results across different countries and continents. So I think it's a very flexible platform where you have people who are quite enthusiastic about the studies and the you know, how can you turn the studies into clinical practice? So I think it's probably a lesson that we're going to take for the next studies. We should always have a group of people, a recognized group of people who have this role of spreading the word and also getting back to us with their struggles, what they're finding difficult while delivering the study, so we can kind of do an iterative development of the study.
James, this term of a data transfer agreement comes up quite a lot. What is a data transfer agreement? And, you know, if I've got a team of 3 or 4 registrars, SHO, and I want to do a project across the country, maybe a couple of the countries, bearing in mind that we had the full support of the NIHR unit on global surgery, and for those who don't know, that's based in Birmingham, it's core funded by the NIHR. It comes with a lot of research managers who help us with the technicalities and navigating through a university, which is really one of the ways we deliver these projects. So would you recommend, if I was doing a project and someone from the United States drops a data transfer agreement, would you recommend that I take that on, or is that a step too far for me as an individual training surgeon?
Yeah, it's a really good question. It's one that's perhaps limited some of the work that's happened in North America, for example, in the past. So traditionally, we struggle with America and Canada in terms of building big collaborations because they often require institution-specific data transfer agreements. And they're effectively agreements between whichever institution is collecting the data and whichever institution is hosting the data and sponsoring the study, to basically agree to some common rules about data security and confidentiality and how the data will be held and disposed of over time. And what we did within this project was to have a set data transfer agreement that we defined at the start in collaboration with our legal office. It didn't take a huge amount of work, and that's something I'm sure we can share an example of, for example. But once we had that data transfer agreement in place, we were quite strict with sites in saying that if there needed to be changes to this or they needed to adopt their own data transfer agreements, then we wouldn't be able to accommodate that at this scale. So if I was starting a project and wanted to work with 3 American hospitals, maybe it would be feasible to think about doing bespoke data transfer agreements, but it might take months on end. But if I was thinking about doing a project at large scale in hospitals that might need these, probably what I would do is to set a data transfer agreement at the start, which is a consensus and would meet regulatory requirements, and then make it a part of the study inclusion that centers have to be able to agree to this without any changes if they're happy to go ahead. Works well for a low-risk study such as this with observational data and anonymized.
And then Lizzie, can we just talk about Redcap a bit? Because people, we run our studies on Redcap and people want to run it on Redcap. And I get a lot of people approaching us about how they can do it, what are the costs, etc. But my question is this. Is it possible for a group of trainees to set up and run their own Redcap, or does a Redcap-based project need dedicated administrative support?
No, it's absolutely possible for a group of trainees to run it. Normally, Redcap is still centered within research organizations. However, the day-to-day runnings of it is easily deliverable with relatively simple training by trainees or anyone who's interested, like medical students as well. It also means that what we were able to do was have quite close communication with our collaborators. So there was immediate feedback. We had a very good sense of the front line issues that people were encountering. And we could directly, because we had a small team that was dedicated and manned this, we're able to adjust it, we're able to pull out things and information if we needed to. So we were able to move this project at each step, despite it being a huge project, face every challenge as it came along, and it was all centralized around the database. So yeah, definitely can be delivered.
So Lizzie, sometimes you need some soft money just to support the Redcap server and a bit of administrative time. As a group of trainees, is it realistic to go out and seek £5,000 to £10,000 from someone?
Yeah, absolutely. There are multiple organizations and charities who will be able to support small grants that will get a study off the ground, just so that there is some database access and perhaps a light touch administrative access as well. You would need to develop your question, know which patients you want to benefit, and then approach the right organizations. But the opportunities are wide and very much available. So definitely would encourage you to seek that.
Thank you. I'm gonna wrap it up there. As with these things, there's loads we could talk about, and there were some nuances of how we got the thing up and running so quickly, the international communication, how we work via regional WhatsApp groups, and the various levels of committee that we had to set up to deliver this kind of democratic project. But we'll have to get into those another time. We recognize this as being a responsive project, that the surgical community, the global surgical community, called out for it, but actually working at those timescales and moving quickly with shorter protocols, and adapting to whatever the local approvals are necessary. So in the UK, most of this is done through audit and service evaluation. Some places internationally, they did get ethical approvals. But having that flexibility around what the approval is, having some flexibility around time is one of the other ways that we've got this up and running so quickly. We are but 4 people of the whole COVID surge team, of which there was a, by necessity, a central management group in Birmingham, but there were international committees and different people feeding in, which is complex, but is the only way that it was able to happen. And we're really presenting on behalf of those people today. So we'd like to thank them very much. And we'd be delighted to hear from any of the trainees who wanted to get more involved in COVID surge and the work going forward as we look to the 5-year vision of what it could be. But for now, thank you and goodbye.
Bye.
Very much, James and Anil and Lizzie and Joanne, and for that video, and for all the amazing work you've done with the COVID Surge project. And it's made such a huge difference to, well, surgical practice worldwide, and it's brilliant to see it have all worked so well. We don't have any questions at the moment from you all. So what we're going to do is we're going to move on now to the next presentation, and then we can pick up questions at the end of Martin's talk. And I'm just so pleased to be able to introduce Martin Landry to you, who's Professor of Medicine and Epidemiology in Oxford. And I've known Martin for at least 10 years, I think, because we had the opportunity of working with him in the NMRC Methodology Hubs project that was done a few years ago. And at that stage, I kept hearing from the Oxford group, oh, we do big trials, all that matters is large-scale trials. And I wasn't really sure then what that meant. And it's just been brilliant the last year, actually seeing that firsthand as he's run the recovery trial. And I've had the privilege of kind of overseeing it in Bristol and being on the receiving end of such a well-organised, incredibly important project, which has been adapting as it's gone all really fast and making such an enormous impact to all of us is just incredible. And in fact, I read up Martin's bio, as you might imagine, in order to introduce this. And what he says at the end of it, I'm absolutely certain it's wrong and it needs to change, Martin. It says here that within 100 days, the recovery trial showed convincingly that dexamethasone reduces mortality by 1/3 amongst the sickest patients. And then he goes on to say, and that was adopted into standard care worldwide within weeks of its announcement. And that's what's wrong. It was adopted worldwide in standard care within days of its announcement. And certainly in Bristol, we literally had a drugs meeting within 48 hours and got it sorted out the following week. So I think you need to put within days, Martin, not within weeks. So I'll shut up now and hand over to Martin, who's going to tell us about how on earth to undertake research that matters to patients, to health professionals, and to the health service worldwide in a pandemic. Thank you very much, Martin.
Well, thank you very much, and it's a pleasure to be with you. I recognise that COVID is a new condition for us all, and many people haven't worked in infectious diseases before. That includes me. Up until the beginning of March, I'd never done any research in the context of infectious diseases, let alone a clinical trial. So it's been something of a learning experience for me. But I thought I'd share some of that experience and some of the background and how we got to where we are now. I'm going to take you back to early March, the first week or so in March, when we were hearing the stories from northern Italy of the devastation that COVID was causing over there, the challenges with ventilation, whether enough ventilators, enough beds, serious rationing, serious stress. And here in the UK, we were looking ahead to the vision of a massively overstretched health service. We didn't know if we'd have enough beds, enough staff, enough ventilators. Huge time pressures on frontline medical staff, not only time pressures, but huge personal stress, given that there weren't any treatments that we knew would be helpful. And then huge numbers of unwell, anxious, elderly, and alone because of the infection control measures, patients. And at the same time, you realize that 1 in 4 of those hospitalized patients will not make it out of hospital alive. For those patients who are sick enough to require intensive care, then that might be 1 in 3 or even 1 in 2. And there was no certainty about how to treat this. A new virus, a new disease, literally hundreds of candidate drugs, many opinions, both from the scientific community, the clinical community, but also in the media, in the social media, and in particular, even from some very high-profile politicians, making claims that certain treatments were miracle cures or others making claims that certain treatments were going to be outright dangerous and should not be used. But all of this was hype, hope, and speculation, and there was no evidence. A few small little trials which became spun out of control in terms of their impact, some inconclusive randomized trials. And it struck me and struck the team working with me, in particular Peter Horby, it was unlikely to be a single big win. This wasn't something like bacterial meningitis, where a single drug was going to essentially turn something that was fatal into something that was easy to survive. This was going to be much harder, and we'd need perhaps multiple treatments with modest effects, as we've seen with HIV, with tuberculosis, with heart disease, with diabetes, and that needs large-scale randomization. Now, this is a work I've I've been working for the last 10, 15 years on the whole area of regulation. If I was giving a talk this time last year, I'd have talked about the advances in data, the need for regulation to allow us to do smarter trials and so on. But I've sort of just picked out this one piece, which is about what a high quality clinical trial looks like. And this was work that actually I was involved in and part led with FDA and others, and came up with a definition that a high quality trial is defined as the absence of errors that matter to decision making. In other words,
Martin, sorry to interrupt. We're not seeing your slides at the moment now. Did you want to? No, it's been. I suddenly thought we must be needing to see this definition. So,
right. Okay, that's a fault in my ability to navigate the system.
It's okay. It's great listening to you talk. Quite happy with it.
Well, I think it's better if you have something to look at that's not me. So there you are. There we are. I will go back to the beginning briefly. Apologies. So I just to give you the context, I've been merrily clicking through these. So this is the beginning of the year, and this is clearly the epidemic, well known to all of us. As I say, the impact on lives, the uncertainty, and the need for randomization. So here we are, quality. What quality in clinical trials, absence of errors that really matter to decision making. In other words, those that seriously impact the safety of the patients or the credibility of the results. And it's not because we want results just because results are nice, or we want a publication in the Lancet or whatever. It was a constant focus on we need to solve a personal patient health question and a public health question. We need to solve the crisis. That was what we were after, was results that would change the trajectory of COVID. And so when you apply that in this context of the overwhelmed health system, the very large numbers of sick patients and so on, here are some key principles. The first was we had to have results that could rapidly impact on clinical care. We had to consider the well-being of the patients, obviously, but also, and you'll never see this in any GCP course that you might do, we had to consider the well-being of the staff and the availability of the resources. So that really drove us to focus only on what matters. Anything extra went. Anything that made it harder to randomize went. So all the usual traditional practices, the extra baggage that so many have added on over the last 20 or more years, we had to examine that and say, is this necessary? Will this drive towards a high quality trial? Built with that was, this was clearly going to have to be a whole team effort. And that whole team, as I'll show you a bit later, has turned out to be truly massive. And it seems to me that in the context where COVID was the one and only issue in every newspaper, in every conversation, and we were trying to do a clinical trials, which to some sort of sounds somewhat scary or used to, in the context of this pandemic, there had to be transparency and openness about everything we were doing. And so on the 9th of March, Peter and I started writing the protocol, and I had on my desk 2 papers. This is the first of them. This was from 1984. Why do we need some large, simple randomized trials? To be clear, this paper never argued that every trial has to be enormous or every trial has to be simple. There were lots of reasons for doing other types of trial. But particularly, look at the last line or last sentence. Particularly, large simple trials can be used to suggest the possibility and desirability the effects on mortality of various widely practicable treatments for common conditions. What was so interesting was when you read this paper, and it's only about 4 pages long, if you replace the words cardiovascular or myocardial infarction with COVID or coronavirus, everything else pretty much slots into place. And of course, that same group did the master demonstration trial of the ISIS-2 trials of acute myocardial infarction in the late 1980s. And this trial came out actually just as I entered the clinical years of medical school, changed the practice of cardiology overnight, and actually changed much of what we do in clinical trials. On the left, the famous one-page case report form. On the right, very clear, compelling results on mortality, showing that aspirin and thrombolysis are beneficial, and if you use the two together, the benefits are additive. Very clear results, change practice, very simple trial to do, but stuck to the scientific principles. But what I like is this paragraph, which is on the front page of the protocol. Again, on my desk as we were writing the recovery one. By far the most important determinant of the success of the trial is the extent to which the responsible physicians and nurses choose to enter their patients. Hence, the extra work must be and is absolutely minimal. How can you make it, the ultimate goal, as easy to randomize to hydroxychloroquine versus not, as it is to prescribe hydroxychloroquine or not? That's where we were aiming to get. And so the recovery trial design is every hospitalized patient who has coronavirus that you would want to treat is eligible for this study. Coronavirus can affect us all. The trial's open to us all. The youngest patient is under 6 months. The oldest is over 102 years. There are 40 or 50 pregnant women, a similar number of children. 18% are from Black, Asian and minority ethnic groups, 60% have comorbidity, 20% are over the age of 80, another 20% are between 70 and 80. It's open to everybody. And the focus was on mortality. Clearly we randomized, and we had a one-page case report form, 1 page at randomization, 1 page at 28 days or discharge. And then, because this is the 2020s and not the 1980s, linkage through to many different routine healthcare data sets. The consent form, as an example, there's the 1-page consent form. I haven't done a copy or screenshot of the information sheet, but it's 2 pages, and it was focused on informing the patient, remembering that these are patients who are seriously sick, alone, and the last thing they want to do is read, as was the case in one pharmaceutical company trial, 8,000 words of small print before signing a consent form. Here's the design, at least the initial design. As I say, patients admitted to hospital with coronavirus infection that you would want to treat with one of these, perhaps with one of these drugs if they worked. And we started off with 4 different treatments. You can get any one of these or no additional treatment. In other words, all the usual NHS care, but not one of these drugs. Lopinavir or ritonavir is an HIV treatment. Dexamethasone, we all know, is a steroid. Hydroxychloroquine, malaria and rheumatological conditions. And azithromycin we think of as an antibiotic, but does have some immunomodulatory properties which might turn out to be useful in the context of this disease. 28-day mortality, the primary outcome, as I said. And here's the record linkage. I mean, I have to say that I never thought this was going to be possible at the beginning of the year. And I've been working in health data for a long time, and particularly around trials. But there's, what, 14, 15, 20 different data sets from different parts of the country, looking at hospitalization data, survival data, intensive care unit audit data, and renal registry data, but also looking at COVID-specific information. We took the principle that if the NHS was collecting it, why would you spend time asking research nurses or clinicians to record it, or bothering patients who remember are seriously sick to ask them yet again information that you already know, the NHS already knows? How can you enhance both the background information you have and the foreground information about what happens to people? We have permission for this for not only for 28 days, which is the primary outcome, but for 6 months, which is a pre-specified analysis, and actually for 10 years of follow-up. And we knew nothing. Nobody had ever heard of long COVID when we started this trial, but it seemed to us we needed to understand the long-term effects of treatments. So this trial started rapidly. On the 9th and 10th of March, we wrote the protocol. On the 19th of March, we recruited the first patient. 8 weeks and 9 minutes later, we randomized the 10,000th patient. In the first 100 days, we randomized essentially 12,000 patients. Every acute trust in the country, from the Western Isles of Scotland to the southern tip of Cornwall, from Northern Ireland across to the east coast of East Anglia, are involved in this trial. Some have recruited large numbers, some small numbers, but it really is a whole nation, whole team, whole NHS effort. And so we got, within those first 100 days, the first results. So on the left, hydroxychloroquine, on the right, lopinavir, the HIV treatment. Remember, hydroxychloroquine had been widely promoted. It was used on a sort of emergency access process in France, in the United States. It was extensively used in Brazil. It was used in India. And at least 3 presidents had actively advocated for this as the treatment, as a miracle cure. Turns out when you do the trial in patients who've got COVID and in hospital, the treatment is completely useless. On the right is the HIV treatment lopinavir, perhaps less famous, but equally widely used. These were first and second choice, or second and first choice, depending which guideline you looked at, in national guidelines, in about a dozen different national guidelines around the world. Based on expert opinion, based on, I don't know, biology or something, turns out not based on fact. The treatments are ineffective and have been abandoned. Dexamethasone, though, the 16th of June, and this, Jane, is where you were talking about earlier on. That was quite some day, I have to tell you. When you know this result and you know that you're about to do a press conference to announce it, and you know that the drug costs £5 for a course of treatment, is on the WHO essential medicines list, is available in pretty much every pharmacy, in pretty much every hospital worldwide, that it reduces the risk of death by a third amongst the sickest patients, by a fifth amongst those who require lesser forms of respiratory support, such as oxygen. That's quite a thing to know and be about to tell people. And one of the things that ticks through your mind, and Peter and I were talking about as we paced up and down the corridor, is you've got to be damn sure you're right. Now, the evidence is very, very clear. Clear evidence of benefit overall, but very clear evidence also of this trend works best in the sickest patients. We announced that at 1 p.m. By 5 p.m., Chris Whitty and the other chief medical officers had written to every hospital saying we think this should be standard of care. And it's one of those occasions where you can say at lunchtime you announce the results. It's policy by tea time. It's saving lives by the weekend. And yes, it did get rolled out over days and actually weeks. It took some time for it to filter through all the way to the WHO and whatever else. But by then, people were doing it. This was something that people could do. And it's still the only treatment known to save lives in COVID. It would not be known if it wasn't for this trial. We actually had people writing to MHRA or Royal Colleges or to the university saying, you should not be doing this trial because this treatment is dangerous. It's dangerous to suppress the immune system when people are fighting an infection. Now, those arguments were well reasoned, but the uncertainty was there because there were also other people using the steroid drug. We've had comments back to us, I don't know why they bothered studying this. We've been doing this in Spain or wherever it is for years anyway. The answer comes, there's uncertainty, randomized, get a clear and compelling result, and then enact on it. And so people in the second wave of the COVID pandemic are benefiting from the experience that and the knowledge gained from those people in the first wave. So here we are. We started off with 4 different treatments. I've told you the results for lopinavir and hydroxychloroquine. It doesn't work in hospital patients. I've told you the results for dexamethasone. It very definitely does in those who require respiratory support. The azithromycin arm stopped a couple of weeks ago, so those results are likely to be out really quite shortly. And in a sense, you could say, well, that's job done. We've finished. But actually, of course, let's recognize we've only got one treatment that treats the sickest patients and reduces risk even in the sickest patients by a third, from say 40% to 28% or something. That still means an awful lot of patients on ITU end up dying. We need more. And so the second wave, of course, we all know is upon us. Here's the recruitment profile. You can see that at one point, over Easter, we were randomizing over 400 people per day. You can see that even during the autumn, we've been randomizing over 200 people some day and over 100 people for the last 6 weeks or so. And earlier this week, we passed 20,000 patients randomized in total. We're studying a range of new treatments, some of which we didn't know anything about before or didn't even exist before. So aspirin down at the bottom there, because of the thrombotic complications that have gained notoriety. But we didn't know that at the beginning. We know it now, so we'll study it. Colchicine, an anti-inflammatory drug, usually used for gout. Actually, I use it for acute pericarditis in some of my patients, again, dampening down the immune system. We know that the immune system is important because of the dexamethasone results. And new treatments, convalescent plasma, Regeneron's version of a monoclonal antibody targeted at the spike protein. Now, convalescent plasma, of course, only starts to exist once you have some patients who are convalescing. And secondly, the monoclonal spike antibody, only when you've actually done the development work to actually develop that as a drug. So new treatments. And over on the right, tocilizumab as an interleukin-6 antagonist, trying to tackle those patients with the severest forms of COVID, hypoxia and inflammation. And just on that tocilizumab, why do we need yet more evidence? Well, basically because the position is not clear. Here's a meta-analysis of all the existing trials that have actually announced results on mortality and given data. And you can see that overall, that's what, about 1,400 patients or so, 1,200 patients randomized, about 150 deaths. It's an uneven randomization, 12% versus 10%. And in fact, what you can see is that there's no overall effect on mortality. Now, each of those trials is pretty small. It's all pretty inconclusive. We know RemapCap have said there's a 1.87 odds ratio for better outcomes, which is a complicated scale, but we don't know how many deaths there are. We don't know how many of those patients were treated with dexamethasone. We don't know what the scale of the improvement in mortality might or might not be. We need more evidence. Actually, in recovery, if you look in the middle there, we already have 1,100 patients versus 1,100 patients on the tocilizumab comparison. The DMC are happy for us to continue. That tells you one thing: this is not a slam dunk. But we also have the largest trials of many other treatments. Convalescent plasma used in 100,000 Americans. Were they the lucky ones, or were the ones that didn't get it the lucky ones? We don't know. We're doing the trial, 3,000 versus 3,000 to date and climbing. The monoclonal antibodies, 700 versus 700. Aspirin, as I say, gone past 1,000 versus 1,000. The colchicine only started about a week ago. But our aim all the time is to get very clear answers. Clear answers might be this treatment works, as we saw with dexamethasone. This treatment does not work, as we saw with hydroxychloroquine. It might even be that this treatment is unsafe. We need clear answers, and that's likely, given the current event rates or mortality rates, to require at least 2,500 patients versus 2,500 patients. But we also have to bear in mind that the benefits might be restricted to a particular subgroup. So if you take convalescent plasma as an example, then it's much more likely that will work in patients who have not or not yet managed to mount their own antibody response than it will in people who have mounted an antibody response. So before we dismiss it as a completely ineffective treatment, we need to be clear that we're not dismissing it on the basis of a lack of statistical power. We need these big studies. A whole UK effort. So what the graph on the left shows is one bar and one dot per NHS trust. Actually, this is just the English data rather than the Scots and the Welsh or the Northern Irish, but the pattern's pretty similar. You can see that some hospitals or some trusts have been incredibly busy and seen huge numbers of patients. This is just in the last 2 months. Others have been quieter. On the dots, though, what you can see is that the proportion of patients recruited is very variable. On average, 1 in 10 patients admitted to hospital with COVID have gone into the trial. That's impressive, but frankly not good enough. If it had been 1 in 5 patients, we would halve the time to finding the answers that we need. And what's interesting is the level of variability. There are some busy hospitals that have recruited, let's say, 25 or 30% of patients. There are some busy hospitals that have recruited, let's say, 7 out of 1,500 patients. Now, I think that that's not going to be down to differences in the local community. It's not going to be down to differences with individual doctors or research nurses. It's much more likely to represent systematic issues within particular hospitals. How does one prioritize research versus care? Actually, the chief medical officers at the beginning of the year said there is no versus. Clinical trials are part of clinical care. That's what it means to tackle this virus. And on the right, I've put a couple of quotes, which I really like. They were sent in by local investigators. The recovery trial has inspired many more junior doctors in our trust to look again at a career in research. It's given an opportunity and access to treatment to our patients they otherwise would not have had. And at the bottom, we're pleased to have taken part and contribute to an effort that helped to provide clear answers. I talked about transparency. Everything that I've said, actually with the exception of the graph on the previous slide, but everything else I've said is available on the public website. Every protocol, every ethics submission, every MHRA submission, every bit of training material, every bit of patient-facing material, every result that's known, all of that is on the public website, along with the overall recruitment on a live basis. It seemed to me that if we're expecting patients to take part in our trial, why would we keep the protocol confidential? It makes no sense at all. Why would it help? Why can't we help others around the world also do trials by making a successful protocol open? Why would one hide the recruitment numbers? Because somebody else might be thinking, you know, I might like to do a trial of colchicine. Is it worth their while, or are we actually going to answer that question and they'd be better off studying something else? Openness is important on all levels. And so just to conclude, I thought it'd be useful to have an American perspective. And so this is from the New York Times. It's one of my favorite articles of the year. There are a few of them actually, but this is one of my favorites. I didn't write any of it, although I was interviewed for it. And I certainly didn't write the title, Where is America's Groundbreaking COVID-19 Research? There's an awful lot to learn from Britain. But what I like about it was that it picked out essentially 6 different areas where we've really made a difference. It was designed to be easy to take part in. I told you that at the beginning. The key was we had to get the science right, but it had to be practical. The second is the protocol was quickly approved. And I was talking to MHRA at their annual meeting earlier on today and gave them enormous credit for that, but so HRA and others, adopted by all hospitals across the country. The patient data has been a key feature for us, so we have no loss to follow up. Leaders in government, chief medical officers and others have really driven trials are part of research and done that over and over again. Of course, the CRN, the Clinical Research Network, and I think really key has been that we have been somewhat successful in saying, actually, let's bring clinical trials to the bedside. Let's take the clinical trials to the patients, and instead of expecting the patients to come to the clinical trials. The protocol's written in a way that you do not need GCP training, you do not need to be a research nurse, you do not need to be professor or associate clinical lecturer or anything else. Every clinician can take part if they wish to, with very brief but relevant training. Oops, if I just finish, sorry about this, I'm clearly incompetent when it comes to the technology. I think it's just helpful to look ahead and think, what have we learned from all this? I mean, first of all, we're not out of COVID woods yet, so there's more to do. But the arbitrary use of unproven treatments really should be avoided. One has to ask oneself very serious questions. Large randomized trials are a critical component, not the only feature, of high quality clinical care, and compelling results absolutely change practice. But for that to happen, the trial must be feasible, it must be inclusive, and it must be focused on what matters. That does require leadership, it requires coordination, it requires collaboration, a sense of fairness, a mission for fairness and transparency. But as I say, that's not only relevant for COVID, that's relevant for the burden of many other common diseases that have not gone away just because C19 has been on the front pages this year. So very finally, to thank obviously the funders, obviously the various partners, but enormous thanks to, I think it's something in the region of 3,500 to 4,000 doctors, nurses and pharmacists and research staff at NHS hospitals across the country. we've endeavoured to list them all in the supplementary appendices of all our publications, but in particular to the thousands of patients. 20,000 patients so far have taken part in the recovery trial. People are being treated today better because of the contributions of patients who contributed in the past. But in COVID, just to conclude, let's remember that we talk a lot about numbers. There's a sort of ticker tape tracker across at the top of every newspaper about the statistics of number of cases, number of admissions, number of deaths. Oh, it's up a bit, it's down a bit. But remember that behind each one of those is a real person, a real family, a real story. And over the course of the last 9 months, roughly speaking, 1 in 4 patients who've gone into a hospital with significant COVID have ended up not making it out of hospital alive. That's why we do the trials, is to find the treatments that work and use them, and the ones that don't work and abandon them, improve care and improve the care for individual patients, their families, and for public health. Thank you.
Martin, thank you so much. Could you stop sharing your screen? That'd be great. It's just brilliant to hear it straight from you. I love hearing it on the news and reading the papers, but to hear it from you as well just adds the whole extra nuanced interpretation of what's gone on. It's amazing. Thank you so much. We've got some questions on the chat here, which I'm going to ask you. And to start off with, I bet you've been asked this one. I bet you've been asked them all before. You've got literally hundreds of candidate drugs. And how on earth do you decide which one to enter in next? And I wondered if you could maybe give us the story behind the culture scene, because that took us by surprise in Bristol.
Yeah, sure. Well, at the beginning, I mean, first of all, we started this trial at risk. We didn't have a grant, and we decided we'd just get on and do it. There's an awful lot to be said for just getting on and doing things. The grant did actually turn out to arrive 2 days later. And one of the conditions was that some of the early treatments would include those recommended by a committee called NERVTAG, which actually Peter chairs, which is a government advisory committee, sort of, if you like, just one step below SAGE is one way of thinking about it, which had done reviews of which treatments might be repurposed treatments might be suitable. In those early days, we were picking treatments that had some reason for thinking they'd work, a known safety profile, enough drug that we could actually do a decent sized trial, and in particular enough drug, or the potential to get enough drug to enough people, that if it turned out to be successful, it could be deployed as a population health treatment. So the fact that dexamethasone was this treatment that's in every pharmacy and could instantly be rolled out was no accident. We'd chosen treatments like that. Now, more recently, over the course of the summer, things have changed around a bit, and a committee called the COVID Therapeutics Advisory Panel was set up, chaired by Patrick Chinnery from the MRC, and is essentially an independent group of advisors. Andrew Mumford from Bristol, a haematologist, is one of them, but there's people looking at thromboembolism, at antivirals, immunomodulation, and 1 or 2 other things, who are reviewing through literally hundreds of suggestions. There's even an open website where people can make their suggestions, as well as a trawl through the past literature. And the culture scene specifically came up through the sort of immunomodulatory sort of subcommittee. It has known properties on the inflammasome in terms of dampening the immune system. Obviously, dexamethasone is also, if you like, a general immunosuppressant. We don't know that it's going to work, but what's been interesting is that although we all think of it as a gout treatment, and we all think of it as being very limited by causing lots of diarrhea and not a treatment that we want, I think partly because none of us actually want gout. It's turned out that actually in the last couple of years it's been used for acute pericarditis and been trialed for acute pericarditis, and turned out we use it for 3 months or more in acute pericarditis sometimes, without much in the way of side effects and with benefit, proven benefit. And actually over the last 6 months or so, perhaps it's slightly longer, there are a couple of trials in acute or recent MI which have also suggested benefit, big trials of 4,000 people or so. So this wasn't a treatment that was completely untested and just sort of plucked out of the air. But on the other hand, how did it win over some other candidates? Well, again, it's cheap, cheerful, and available. And when one thinks about a global pandemic, there's an awful lot to be said for that. And we've seen already that the cost of the drug does not necessarily equate to the value of the drug.
No. Fascinating. Watch this space. So every week in our COVID committee in Bristol, when we know the next results are about to come out, we have a sweepstake, and we wait to see what happens. We've been wrong every time, so we wait to see. Another question.
We've all been wrong every time. I've been wrong twice. Once is I've been wrong on what the drug's actually going to do, and the second is I've actually been wrong on when the results are going to come out.
Okay.
So don't take any notice of me.
Next question. It was obviously brilliant that dexamethasone was introduced into clinical practice really quickly and effectively. How could we learn to better disseminate results from trials immediately and get them taken up? What would you suggest?
Well, I suppose it does depend an awful lot on what the balances are. I mean, here we had a condition with a 1 in 4 mortality, and a reduction in that mortality of a third. So it was a very big result on the ultimate outcome where there is no alternative, and a drug where we knew the side effect profile. I mean, everybody learns at medical school the side effects of steroids, particularly in lowish dose in short term, and everybody can deal with the hyperglycemia that might or might not occur from time to time. So that was one which was relatively easy to get into practice, and I think there probably are some things we'd learn. We wouldn't have done it quite as fast, I think, any of us, if it had been for a chronic condition. But it was very definitely the right thing to do. Yeah, we couldn't sit on those results for longer than about 5 days or whatever that we actually were. We did know. You know, I knew the answer for about 5 days before everybody else did. You can't sit on a result like that for any longer, and hence the press release, and then the Med Archive as quickly as we could. And then the preliminary publication and a final publication will come out soon. I think it would be a bit different if what we had been studying had been an entirely novel drug which required significant funding, or which had particular issues around safety profile or whatever, or where the gains were less obvious. And if one looks at something like convalescent plasma, then obviously the challenge is not only once you've got the result, but how are you going to get enough of it? If it was a really significant benefit, how are you going to get enough of it? And so the first challenge is going to be, how do you scale up, if you like, harvesting, or for a drug, how do you scale up production before you go down that route? So there are reasons why some things will be slower, but I mean, I think there's a lot of things where actually what one's doing is studying 2 drugs that are widely known and widely used, and is my flavor of heparin better than your flavor of heparin? Ted Stocking's worthwhile or not? These are all the sort of questions which really could be rapidly implemented. But what often delays things is that the answers aren't clear. You have answers that people debate, and they start to then unpick the subgroups and the secondary outcomes, and they sort of look at the data sideways and whatever else to either try and interpret that something definitely there is a signal there, or there isn't a signal there, or according to their prior beliefs. Usually, if you get clear enough results, you will change practice, because it's very, of course there will be extremes who will say always, I told you so, and there'll be people who said, I just don't believe it. But you'll sway the vast majority if you get clear results. Clear results, not just to pass a regulatory hurdle or a NICE hurdle or a commissioning hurdle, but a guidelines hurdle, an individual doctor and patient hurdle. You've got to be looking all the way to the end, to the health benefits, not just the next step about, do I get it published or do I get a license?
Thank you. And large trials help get clear results. Lizzie's waving her hand. There's a load of you're not talking to me. No, sorry. Next question. There's a few questions on the chat about the governance and the ethical processes that were obviously simplified for recovery, and how on earth do we maintain that beyond the pandemic?
I mean, huge credit to all involved, particularly in the early days. And we went from protocol, written on the 10th or so, and approved within about a week. That's not sustainable. A lot of people did not sleep, including the regulators and the ethics committees and everybody else did not sleep for weeks on end during that phase. One cannot continue in that way. It's not healthy. One will never recruit another regulator if that's what it means. So I think that But I do think one of the first thing I noticed was when you empower people to say yes, as opposed to dither, delay, ask more questions, refer it back or refer it on to the next committee, it's amazing how people are perfectly capable of making sound, reasoned decisions. And it's not clear that those decisions would be any better if you've gone through all the rest of the elaborate processes. We didn't skip any steps. We were challenged. There were things we were asked to change or things we had to have a dialogue with the MHRA or the ethics committee or whoever. But all the time, people were thinking, how can we make this work? As opposed to somehow trials are risky. Clinical care is risky, particularly clinical care in the absence of evidence is risky. So the question is, every little burden that you put in the way, is it really justified? That delegation of duties log, I mean, why? I mean, do you have a delegation of duties log when you're a consultant surgeon, you have a team of surgeons, of junior staff with you? If you run a ward as a nursing sister, do you have a set of delegations? You have training mechanisms in place, you have support mechanisms, you have mentorship. That's part of what these organizations have. Why do you then need to suddenly document everything in delegation of duties logs just because you're prescribing something by the toss of a coin? In other words, you're prescribing something to half the patients as opposed to prescribing something with no ability to learn to all the patients. It makes no sense.
Yes, I really hope that's the case. And just another couple of questions. I think we're all right for time. We're allowed to use up the coffee chat time. Go on, Lizzie. Liz wants to comment.
Can I ask, I was on mute. Classically, everybody at least has to be on mute once during every one of these, I'm afraid. Professor Factory, just on your point that you just mentioned, so you alluded to the fact that your protocols are all online, very transparent, and our team have very eagerly seen them as well, and they're very streamlined. And you mentioned that you've really cut down and made it very clear for the information for patients. Do you think this is something that we can carry forward, especially in things like drug trials?
Yeah, I think it's something you have to carry forward. I mean, take dexamethasone. We didn't need to study for adverse events and even for serious adverse events. I mean, notwithstanding the fact that a patient admitted with COVID and on ITU is essentially one infinite series of adverse events happening second by second, and it's only a question of which ones you write down. I mean, that doesn't make any sense either. But we know the safety profile. We know lots about dexamethasone. The only thing we didn't know was does it save lives in COVID? So that's essentially the only, not quite, but that's the thing that you focus on. It's different with other drugs. You may not know so much, but you don't have to learn everything in every trial. So if you take the Regeneron's monoclonal antibody, which is unlicensed, it actually now has an EUA for outpatient use in the States. We don't know the full safety profile of that. But again, the purpose of the recovery trial is to understand, does it save lives? There are other phase 2 or phase 3 trials which are much, much smaller because they're not going to study mortality, but which are collecting information on adverse events or serious adverse events. And even in recovery, we have added particular targeted questions, particularly to work out whether there is the equivalent of transfusion type reaction. So as the infusion goes in, does somebody get an anaphylactoid type reaction? Does their blood pressure drop, and so on and so forth? So you've got targeted questions. That's something which we do want to know about, but we don't need to know everything in every trial. And there wasn't a pre-written protocol for this. We didn't take a trial unit or sponsors or something pre-filled template to write this. We started out by saying, what do we need for this trial? And so you won't find the 3 pages of standard definitions that don't make any sense of adverse events and what's related and what's not. It's not in there because it's not relevant. So very much focused. And I guess if the pieces of protocol I'm most proud of, I think it is worth having a look at the sort of quality by design type pages, the approach to monitoring, the approach to training type pages, because they were very deliberately written, thinking, well, we know there's these requirements, we know there's these GCP principles, we're not trying to break those, but let's have a sound reasoning to justify or explain what and provide the rationale for what we are doing. And so there's only a couple of pages. It's not written in the sort of language you'll see in most protocols, but I think is very useful as a sort of model. Do not copy and paste it. That's the whole point. take it, think about it, and apply it, the principles to your own trial, because that's actually how you'll drive up the quality. So you know, I think those are the bits as I was writing it. In some ways, I enjoyed writing the most. I mean, you know, it's a randomized trial. It's got outcomes. It's got eligibility criteria. I mean, yeah. But those are the bits I guess I enjoyed writing the most.
I'm going to start worrying about you, Martin. Two more quick questions. So I think trainee involvement in recovery and trainee involvement in COVID surge, which is the other project that's been on tonight. Both of you could answer that one. Is there how are you doing it in recovery, or how you did it in COVID surge?
Perhaps we can talk a little bit about the COVID surge trainee involvement. As with many of the sort of National Research Collaborative projects, there's trainee involvement at every level, really embedded within the group. So within the core steering group, the dissemination committee, you know, there's a medical student in Morocco that's done just a wonderful job of getting hundreds of hospitals involved in the study, trainees involved in the protocol phase. And I think it's many of the lessons that you share with us this evening, Martin. You know, everything is very streamlined, simple. You know, we've got short CRFs, a short protocol. Our protocol was 3 pages. I know it's not a trial protocol, but we did our best and the front page is just a letter to invite people to get involved. So it's the shortest protocol we've ever been involved in. I think it's all about creating a culture around trainee involvement in research. And I really liked your reflection from one of your local principal investigators about how they'd seen this powerful research, you know, getting people excited about the research process. And I think, you know, as a group, and we should absolutely be encouraging that wherever possible. Lizzie or Joe, do you have any reflections from dissemination committee perspective?
Yeah, definitely. As you said, trainees played a very important role, and actually they've taught us how to disseminate the results all over the globe with different strategies in different countries. So, for instance, we have 2 trainees leading Brazil, and their strategies is, for instance, to translate every piece of text we need to communicate to the collaborators there, and then obviously we have the visual abstracts of each paper we publish, and they also want to translate them in their native languages. And then we've been really learning from the trainees because they're very close to the surgical community, and they're very close to what we can actually change in clinical practice. And the trainees have been a huge workforce behind not only setting up the study, but also disseminating the results in a very efficient way that can actually change practice.
And Martin, you've got the Associate PI scheme now in recovery, which is only just opened, I know, but apparently 70 have signed up.
70 have signed up already, which is fantastic. But I mean, all the way through, I mean, actually, if you look at the protocol, it even talks about medical students and clerical staff and so on being part of the trial and contributing to the trial. My son's housemate was in his first 6 months job, or perhaps his second 6 months job as a houseman, and you know, his name's on the paper. You know, because he in Croydon or somewhere contributed to the way that the trial worked there. It gives experience that actually research is not something, and trials are not something that are done by special people, and they're done elsewhere. They're done here and now as part of the clinical care, and that's so important. So you know, we have to make it easy for that to happen, given all the demands on those people. But that was very much part of the intention.
I was very delighted to see, Professor Landry, that on your New England Journal of Medicine paper, you know, it's just a single corporate author group, and we sort of live and die by that as well. And I think it's such a positive way of engaging people across all training stages to make everyone feel like they're part of something bigger, perhaps. So yeah, I think that's a great model for everyone to consider using into the future.
One last question, Martin. So your 28-day mortality, clearly really important, but and you're going to link it to the HESD, to the long-term outcome, or whichever data set it is, to get the long-term outcomes in terms of long-term survival. When will we be hearing those results, roughly?
Well, we announced the dexamethasone results in mid-June, so we're just about at 6-month follow-up for the final patients. Perhaps not quite, because some patients were still in the trial just as we sort of closed it to enrolment. So my guess is that 6 months will be up for everybody in January, and then we have the challenge of writing that up. I have to say that the writing up is one of the biggest challenges, because the papers have come so thick and fast. And we're doing, preprints, press releases, preliminary publications, final publications, and dealing with all the rounds and rounds with reviewers and so on. And it's entirely possible that other results will come up. But yeah, the 6 months will be up in January, and as soon as we can do it after that, we will. There's not inconsiderate. There are considerable data management challenges and all that as well, and that's been a huge learning curve, but I think will be useful to all of us in the future.
Thank you. I'm going to have to shut up because we've gone over time, but thank you so much again to Martin and to James and Joe and Lizzie. Thank you. I'm going to hand back to the to Emily, who's going to close it, and then we'll finish.
Thank you.
Thank you all so much for this excellent session. It's been a real privilege to be able to learn from all of your experiences.